Welcome everyone to our Research and Collections virtual seminar series. It's a pleasure to have you all here with us today virtually. And so just a few logistical things, a reminder, if you have any questions, please put them in the Q&A and then we will ask them at the end of the talk. And so with that, I'm going to turn it over to Dr. Xiaoming Wei, who will introduce our speaker for today.
Thank you, Allison. Let me take a few minutes to actually first introduce about the Channel Island exhibit. This is a initiative that is recently launched, and we are going to actually produce an in-house exhibit in about 2 years. Amy Gussick, Greg Pauly, and myself from RNC, plus several folks in the exhibit department will be heading this effort. So we are at the brainstorming stage. This is the reason that we're inviting our speaker today to talk about the island fox. In the coming weeks, there will be more. So there will be several exhibit-related talks. So today's speaker is actually closely related to the subject matter that we have in mind. Okay, so let me introduce the distinguished speaker, Professor Robert Wayne from UCLA, and also my fellow carnivore colleagues. Actually, for those of us who go back more than 20 years in the museum, we will remember the exhibitions of cats and dogs. Remember that? Those two traveling exhibits actually are co-curated by Bob Wayne and his wife, Blair Van Valkenburg. So actually, Bob is a really close colleague here and has contributed to the museum program quite a lot. And those two exhibits actually are really popular as well. They travel quite a bit. Bob has a bachelor and then a PhD from Johns Hopkins universities, and then he started off as actually a regular morphologist, and then he saw the light, and then turned into a molecular person. And of course, his accomplishment is really long, and I can't really dive into the details, but I will mention two things that are actually really quite popular. One is the red wolf. Bob is actually right in the center of the controversy of the status of the red wolf. And I think Bob takes the line of the gray wolf and coyote are the ancestors of the hybrid ancestors of the red wolf. And the other aspect of Bob's really works that are internationally known are the origins of domestic dogs. And along those lines, Bob's and his team has actually done a lot of ancient DNA and as well as genome-wide works on all sorts of dogs and also wolves today. So he was really well known in those subjects. But today he will be talking about island fox, which is another area that he has started very early. So without further ado, so let us welcome Bob Wayne from UCLA.
Thank you, Xiaoming. 20 years ago, my God, that's a long time. I have to say, but due to some therapy I've had from Regeneron, I feel just as good today as I did 20 years ago. Actually, I haven't had that therapy. It's odd giving a thing virtually. I see recognize the names of many of you. I really miss meeting people in person and interacting with them. So it's a shame, and I hope we get a chance in the future to meet. I'd be happy to talk about anything I present today to any of you. Just email me. I'm going to talk about a more general topic than just the foxes today, and that has to do with extinction and decline in very small populations. Islands, as it turns out, are a great model for how species might exist in the fragmented world of the future. And so how they can persist, and the island fox is an exemplar here, persistence for thousands of generations at very small population size. And that is a model for the mainland insularization. So I'll talk about foxes to begin with and their genomic pattern. I'll contrast them with another island species, or not species, but island population, the Isle Royale wolves in the middle of Lake Superior, how messed up their genome is. Big patches of their genome have no variation. The sawtooth pattern is characteristics of highly inbred populations. And inbreeding leads to congenital defects. And by the time the Isle Royale wolves went extinct, they all had some kind of congenital defect. So just as my talk outline, I'll give you a little bit of to begin with, a conceptual framework, why islands are so important, and that relates to the small population paradigm. Actually, it's kind of the island paradigm in a way. And I'll talk about what is sort of the evil twin of adaptive variation, deleterious variation, how it accumulates in small populations. Many of us think about adaptive variation, how important it is, but actually deleterious variations is orders of magnitude more common in the genome. It's the principal sort of mutation that occurs in the genome and accumulates. Adaptive variants are far rarer. So I'll begin with, as an example of deleterious variation, island foxes. They're small population survivors, and drift and selection have acted on them, and we can model that process. Then I'll contrast them with the Isle Royale wolves, who are major losers in a big way, and they went extinct essentially, but they were founded over 70 years ago. So to begin with, just a little bit about the conceptual framework. Well, we know fragmentation is a huge problem across the globe, and we have really good evidence that fragmentation drives extinction. This plot just shows vulnerable, endangered, and critically endangered species have generally higher levels of fragmentation. And the fragmentation leads to small population size, and then through stochastic or genetic factors, those little populations tend to go extinct, and over a period of time, all the populations that are small will go extinct, and eventually the species is extinct over large parts of a landscape. This leads to something that's often called the small population paradigm, and it has been kind of a leading conceptual framework for at least conservation genetics for roughly about 40 years. And the notion is that this fragmentation process leads to small isolated populations, and that those populations then have become inbred and suffer inbreeding depression. They lose genetic diversity, which is necessary for adaptation. And this then further reduces the population size and becomes even smaller, and then all these effects become more extreme, and eventually the population spirals into extinction. And one has to mention also, there's all sorts of stochastic factors that act on small populations in concert with genetic factors, leading to this extinction vortex. So this kind of paradigm predicts that most small populations will go extinct, and that inbreeding depression will be a principal factor in their extinction. And we have great examples of this kind of inbreeding depression. Here's a few. Florida panther actually stopped reproducing in the 90s, and the males had cryptorchism, the testes didn't descend, the females, and sperm problems. Females had a suite of phenotypic difficulties, holes in their heart, and the population failed to thrive until 6 females from Texas were brought in. They reproduced once, and they rescued the population. It increased, and it's still maintaining its numbers today. Other populations, snakes, the Swedish adder, suffered inbreeding depression, reduced fertility. It's a classic story of the greater prairie. Chicken, reduced fertility, rescued by individuals from other populations, and then Isle Royale gray wolf that unfortunately was not rescued, and all the individuals eventually suffered congenital defects, and the population went from a high of 50 to no individuals over the course of 70 years. However, there are small populations, presumably, that we have no inbreeding depression, no visible inbreeding depression, and that we can learn lessons from. And the island fox being one, the Chatham Island black robin another, golden hamsters, and a Mauritius kestrel, all examples, and there are at least a dozen well-studied examples of no apparent inbreeding depression. So we set out to try to understand how these small populations can provide a lesson that allow us to diagnose, predict, or reverse inbreeding depression in these small populations, which are the destiny for many species into the future. Jacqueline Robinson, my former student, now at UCSF, and Kirk Lallmauer, professor at UCLA, who's a leading figure worldwide in the study of deleterious variation, how it contributes to population decline through the use of demographic simulations. Chris Crisais, who is my student, shared with Kirk, who leads simulations with this wonderful program called SLIM, which is a combination of like a PVA and a genetic analysis. And then Diego Ortega del Verrocho, who was at UCLA, is now heads a group in Mexico, does ABC modeling. So let's start with the island fox. What a wonderful little species. They really are, I've handled about 22 different wild species of canids, from wolves to wild dogs to foxes of various kinds. And they are the most delightful ever. I mean, you can hold them in your lap and they don't apparently mind being handled. They're trap happy. They jump off your lap and they'll go into set traps that you've reset. Often we'd have 50% of our traps would be animals we had trapped the day before. So they seem to have lost fear of humans. They're notorious for stealing sneakers and things out of people's backpacks that they put down even for a minute. Their history is a very curious one. There are 6 populations on the largest islands. They range from 300 to 2,000 individuals per population on the largest island. They probably were colonized on the order of 8,000 to 9,000 years ago. There's a little fossil record, but fossil records will notoriously underestimate the age of first colonization. Paul Collins studied them morphologically. He's the first one that really took a systematic view of them and thought that maybe they arrived there before humans 12,000 years ago. But his reasoning really was not so much the fossil record, but that it took a long time for them to decrease in population size. The island foxes are about 2/3 the size of the mainland gray fox, the classic island dwarfs, like the mammoths. They have extremely low diversity, and it appears that the northern islands were colonized first, and the southern islands were then colonized later. And it's very likely that Native Americans had a role, at least in bringing foxes to the southern islands. These channels are just too deep. It seems very unlikely that foxes could have swum there. Maybe they could have swum to the northern islands, but most people now, I think, believe they were brought to the northern islands too from the mainland by Native Americans. But mammoths made it. It's just unclear whether foxes could actually swim that distance. The foxes, although having this thousands of year history, they have suffered declines, recent and probably in the past, and remarkable decline on the northern island when golden eagles took up residence, and they weren't there historically. Bald eagles were there historically and probably kept the gold eagles out. Bald eagles are principally marine predators, but the golden eagles are terrestrial predators, and they ate all the foxes, basically. And this is really dramatic. My student was studying the foxes and their ecology in Santa Rosa. He went to the mainland for 10 days, a little break, came back. All his study animals were gone. And they were degloved. Their skin was peeled back. It's classic indication of bird predation, of eagle predation. And they were golden eagles. And they subsequently declined all across the 3 northern islands. And critically, the golden eagles generally ate piglets during part of the year when the piglets were of sufficient size, but as the piglets grew up and they couldn't catch them anymore, then they would switch, they would prey switch to foxes. So the Park Service came in, removed the eagles, removed the pigs, and took the foxes into captivity, bred them. They bred very well, reintroduced them to all 3 of the northern islands. On the southern islands, the Catalina population almost been extinct, like 90% of the foxes disappeared on the southeastern part of the island, not across the isthmus, through canine distemper that was introduced. But they did come back from the survivors and some translocations from the end of the island that wasn't affected, but a very dramatic decline, more than 90%. The remarkable thing is that after this happened, 4 of the island populations, Catalina and the northern islands, were listed, and they were bred in captivity. And just look at how they declined in 2000, just handfuls of individuals. And then they bred them up in captivity, reintroduced them, and many of these populations are approaching the size here, here, and here that they were before the decline. It turns out it's the fastest recovery of any mammal under the ESA. Remarkable, and suggests that at least genetic variability doesn't have a lot to do with their ability to recover very quickly. Nonetheless, we went out and surveyed genome-wide. And this is a kind of new analysis that is becoming more frequent, where you actually look at whole genomes. You do low coverage or high coverage sequencing of the entire genome, all 2 billion base pairs of individuals, rather than just a subset or a few thousand base pairs. These are complete genome scans. So let's take a walk through the gray fox genome. This is a level of variability, of kind of heterozygosity. And you can see gray foxes from the mainland pretty high. This is one individual, right? And then Santa Cruz, which is a pretty big island, you can see as a fraction, maybe 10 or 20% of the variability of the mainland gray fox. And so it's persisted under very low variation. This is before the northern bottleneck. Now look at San Nicolas Island foxes. Almost no variability. We called it genomic flatlining. And just a remarkable absence of variability, but nonetheless, they have persisted for thousands of years. This is a comparative diagram of variability on the x-axis and the frequency of that kind of variability across over 100 genomes that have been done. And here's San Nicolas foxes. They're several orders of magnitude smaller in heterozygosity. They're the least variable population known of vertebrates or any of the species, even invertebrates that have been surveyed. But on the other hand, San Miguel and San Clemente, they're more like the Yangtze River dolphin or the African cheetah, still very low. It's not really to get the gray fox where these are the ancestral levels of variation. So all the island populations extremely low. So the genome has very little variability where there's a lot of deleterious variation in the genome that might contribute to genetic load and decline, population decline. And so here we looked at a few samples. We had this 1988 sample that I actually captured with some help from my students and colleagues. And then we had a sample after the bottlenecks that I've been talking about. And we actually wondering if we don't go back nearly 100 years, whether there had been population declines that are recent that have attributed or caused this lack of genetic variation. And this just shows the answer for at least neutral heterozygosity is not, actually variation hasn't changed. It's always been low. And it's pretty low here. There's even a kind of a pattern. And then the gray fox from Northern and Southern California have very high heterozygosity that you can see. So how do we measure deleterious variation? Well, there's this wonderful thing if you have whole genome data. You can actually look at genes and you can look at spots on codons, the 3 base pairs that determine an amino acid. And you can ask, well, if at one of those sites, say the 3rd base pair in the codon, can there be a change that changes the amino acid? And if there's every time you change from the 4 nucleotides to another, there's a change in the amino acid. We call those 4-fold sites. So every change from A to T or C to A causes a change in amino acids. So those are all functional. But in contrast, there are what we call 0-fold sites. So that means any change in the nucleotide, that position doesn't cause a change in the amino acid. So those are presumably neutral. So selection can act on these kinds of sites, but not these. So a measure of how many how much fourfold heterozygosity there is a measure, in a sense, of the burden of deleterious alleles. So we look at that ratio. The higher this ratio, the more burden there is. So you can see San Nicolas, higher burden, all the islands, higher burden of deleterious variation. Okay? So that's a bad sign. So then the trick that we can use, it's not really a trick, it's a wonderful tool. We can be more precise. We can make a prediction and ask the question, is all this deleterious variation simply as a result of demographic history? That is, can it be entirely explained by small population size and the sequential founding of all the island population? And these models can be very specific. We can start out with a mainland population, and then we can say the islands, the northern islands were founded and give that an effective size. And then say, finally, we found, say, San Nicolas Island, and that founding size is very small. And then we have a final effective size on San Nicolas Island. That's the demographic history we're simulating. So we're not making assumptions. We're actually asking for a specific demographic history. Can it explain the amount of deleterious variation we find? Here's the San Miguel model. In this case, there's only one bottleneck, because the northern islands were founded once, and then we actually infer some population declines in the mainland gray fox that we then model from genetic data as well. And so I just want you to see that in gray here is the predictions of this model for San Nicolas Island foxes. So it actually models it pretty well. And because the population sizes are so small, there's a lot of stochastic variation. In contrast with the higher effective sizes of San Miguel and the other populations, the modeling is spot on, basically. So yes, the amount of deleterious variation we observe is consistent with demographic history. And now we can explicitly treat demographic history. We don't have to make presumptions about what's happened to our populations in the past, or whether they're steady state or declining or increasing. And the gray fox also very much on. So they've accumulated deleterious variation. Then we can ask this other question. One curious thing about San Nicolas is this flat line that I mentioned has some bumps in it. There are places where the heterozygosity at that spot in the genome is almost as high as the gray fox. So we wondered, hey, that must be selection. And indeed, some of those spots contain great things like olfactory receptor genes, immune-related genes, and we built our whole story that selection is preserving this genetic variation, just not all the way across the genome. But we don't know that's the case until we consult demographic history. It's like looking at a fossil record. If we had it, we could reconstruct the population history for San Miguel this way, and for San Nicolas, and ask, are these bumps really expected? When we count up the number of bumps, is it predicted just from random factors? And the answer is, unfortunately, yes, it is. Gray fox, San Nicolas fox, we applied our models, and this is what we observe. The model predicts this distribution of the number of heterozygosity peaks here, and what San Nicolas Island has is right in the middle. So these peaks are just the result of random factors and demographic history. So given the accumulation of deleterious variation, we wondered, in a sense, can it be entirely explained? Are we missing something, or is there the appearance of these deleterious variants in the skeletal anatomy of island foxes? And actually, inbred populations typically have this kind of congenital defects in the coccyx or in the vertebral column that one can record. It's very common. And so this is actually a reasonable proxy of inbreeding depression. So we set out then to actually measure pathologies from museum collections in a wide variety of foxes from different islands and the gray fox in the mainland. And here's San Nicolas. Here's San Nicolas. And you can see that the pathologies that we find, congenital pathologies in the skeleton, the postcranial skeleton, very low. In fact, lower than the gray fox. And most of them are normal. If we contrast with Isle Royale, wolf, which we know is highly inbred, you can see that it's a very high degree of pathologies. In fact, by the time the end of the population was coming to an end, all the wolves had pathology. So this suggests that island foxes are very healthy. They're survivors. Well, why is this happening? One explanation is that if we view deleterious variation that might cause pathologies in three groups, in strongly deleterious, and the skeletal variation would be the strongly deleterious one, the skeletal problems, moderately deleterious, and weakly deleterious. And we can model this process. We can say, well, here's the distribution of all the mutations in the genome, and we can say a fraction of them are strongly deleterious and some are moderately deleterious. This is called kind of the selection landscape of the genome. And there are various studies that kind of give us an approximation of what these categories should be, especially in humans. And then we use our demographic models, and we can ask, in a sense, does demography alone predict what might happen? And is there any evidence of purging of strongly deleterious alleles? So this is not that important. This is an additive model where alleles are interacting, so the heterozygote may be intermediate. This is recessive, and most strong deleterious genes are recessive. That is, they don't affect the phenotype unless you have two copies of that deleterious gene. The heterozygote is the same as the wild type homozygote. So those recessive, and they're hidden in the genome. And in big populations, when a new mutation occurs, if it's strongly deleterious, it's by definition heterozygous. And it has to be crossed with another individual with the same mutation, very rare in that population, to be expressed and acted on by selection. So in a large population, that almost never happens because the frequency is so rare. So if we look at the mainland, there's a lot of recessive deleterious genes, and in the islands less so, as if selection has knocked out a lot of those strongly deleterious genes. And you can imagine in a small population, there's more inbreeding, so there's more chance to express in the homozygous condition these recessive deleterious variants. Less so in the moderate case, and in the weakly deleterious case, there's no difference. So it suggests that selection is acting on these strongly deleterious variants and reducing the genetic load, in a sense, of these strong ones in the island populations. So we wanted to be really kind of comprehensive about this and not assume one model. So we assume various models of how the islands were founded, and one with inbreeding, serial bottlenecks, and etc. And we looked across all those different models and the mainland. And so here, number one is the mainland. And you can see under all these models, the total number of recessive deleterious variants are reduced. And if we just look at moderately deleterious and strongly, weakly deleterious ones, you can see there's almost no difference between mainland and island. So what we feel has happened is that because the populations are really small, so the probability of deleterious variants being in a homozygous condition and being acted upon by selection is much greater because there's some inbreeding. So you're breeding individuals with this very similar genomes, and these recessive then variants can be exposed to selection in the homozygous condition and purged. And we feel that's probably may have, at least genetically, helped save the island fox. Seems that other populations similarly may experience this kind of purging, and we have begun a genomic study of the western scrub jay. Here, these two forms are dramatically different, the island jay and the western scrub jay. The island jay now just on Santa Cruz Island. They're beautiful birds. They're bluer, they're bigger, which is part of the island syndrome. Birds often get a little bigger on islands. Anything under a kilo gets bigger, whereas the foxes get smaller because they're more than a kilo. And their whole life history is different. And they probably have been there, at least if we accept this divergence in mitochondrial DNA as a clock, they've probably been there more than 100,000 years. So they're long-term survivors on the islands. And if we think that they're really there for more than 100,000 years, all the 3 northern islands were connected at various periods, beginning about 14,000 years ago in one super island. So if they were there then, they were probably on all 3 of the islands, Santa Rosa, San Miguel, and there the lesson is they've gone extinct there apparently. There's not much of a fossil record and only remain in Santa Cruz today. And that suggests that their situation may be perilous, especially given the use of Santa Cruz Island and the introduction of non-native avian diseases. So we're hoping to find a similar result there, that there's some purging, that genetically they're very healthy, and our initial genomic studies suggest that's the case. Let's just jump then to another isle population as a contrast to the island fox. And here, this population on Isle Royale National Park, it's a biosphere reserve, beautiful untouched wilderness in Lake Superior. And it seems like wolves have been able to migrate there on the occasions when the entire lakes gets frozen over. And that began in maybe the late 1940s and founded just by maybe 2 to 3 wolves, but 16 generations ago, and reached a population size of 50 individuals, and then went extinct eventually, just probably a few years ago, through severe inbreeding depression. They weren't reproducing at all. And it turns out this is one of the most well-studied wolf populations. Several papers in Nature about them. The wolves eat the moose, and the moose graze on the trees, and you can actually look at tree rings and see how abundant the moose were relative to the wolves. And then it's a simple predator-prey system, wolves and moose, and you can follow the ups and downs of the moose population and the wolf population. So here are the wolves, and here are their numbers, and eventually they got to 50 individuals, and the moose population declined, and then the wolf population crashed due to the introduction of parvo to the island, and then they maintained low numbers thereafter despite, just gonna close my door, hold on. Yeah, that's better. They maintained low numbers despite the increase in the moose population. So that was a real puzzle. And those low numbers kind of remained for a long time. This wolf immigrates from the mainland in an ice year, and all these little diagonal triangles here indicate when the lake froze over. So the lake froze over, a wolf gets across, and then there's a little bit of a bump up, but then there's a decline to the extinction today. So a really interesting history. So what does their deleterious variation look like? It turns out in this brief period, just a few generations, there's not much accumulation of deleterious alleles as there were in island foxes. In fact, there's not much difference between the variants that we can classify as damaging. And this is a measure of how conserved these variants are. It's actually an evolutionary experiment. Do we see that mutation in any other species? If not, it's probably really damaging. So just to compare these graphs, damaging and benign, not that much different in the proportion of genotypes in regard to heterozygotes and even the total, not that different. So there seems to be a different situation here. They don't have a lot of deleterious variants relative to the mainland, but nonetheless, they show incredible incidence of congenital defects. And here's just a comparison of Isle Royale to Scandinavia, where the wolf population is small and somewhat inbred. Finland, same thing, some congenital defects. And then historically, large outbred populations, no congenital defects or not detectable. And this is just sort of congenital defects they found: extra vertebra, extra ribs, asymmetrical vertebra in Isle Royale wolves. Nice little paper on that. So now let's take a look at the genomes of wolves worldwide. And here's some, we'll start with populations which are historically very large. And this includes the Chinese lowland wolf and Minnesota wolves, historically large across North America. And then populations historically small in the Tibetan Plateau, which was probably limited in area for some time in the late Pleistocene. And those wolves have lower genetic variability, also isolated. And the Ethiopian wolf, actually a different species, just isolated in the Ethiopian highlands, about 3,000 meters, Again, the variation very low, the lowest of any wolf except Isle Royale wolves. And so, but they persisted for tens of thousands of years there. They're actually a very distinct species. They eat mole rats that live in the high altitudes. The Mexican wolf, species that went extinct, or not species, but a subspecies that went extinct in Mexico and parts of the southern US about 40, 50 years ago, and was brought into captivity. Its genome, very average heterozygosity, very low. But look at this genome. It's like this sawtooth pattern. And there are large areas, whole chromosomes, which are monomorphic, no variation. This is what happens with inbreeding, because offspring inherit two versions of a chromosome that are the same, because they are from parents which are inbred. And so in this sense, these large areas indicate inbreeding. In fact, we can measure inbreeding by how large these runs of homozygosity are. And it's a much more accurate estimate of inbreeding because it's like realized inbreeding. Whereas if we try to calculate it from the pedigree, we really don't know at the individual level how devastated their genomes are from inbreeding. Here are Isle Royale wolves, same sawtooth pattern, whole chromosomes, intense inbreeding. And you can see this kind of a pile up on these runs of homozygosity at this very high level. So the genome is dominated by long stretches where there's no variability. So what does this mean? Well, if we have no variability in these long stretches, that means all the deleterious variants in those long stretches of the genome are expressed as homozygous. And so that's the deleterious variation is being manifest in these long stretches of homozygosity. And in Isle Royale wolves, in Mexican wolves, that's happening to a great extent. We might have 50 or 100 deleterious variants of various effect in these runs of homozygosity that suddenly are being expressed in the phenotype because they're homozygous. So how do we approach this situation? Why are Isle Royale wolves having a problem and island foxes not? Again, it's a demographic history question. We can imagine that some time ago, there was a large effective population size, and that in North America, that decreased to about 17,000 individuals. In Tibet, much less. So what we want to ask is, well, does the smaller Tibetan population size, because there's more homozygosity, purge these deleterious variants? And so this is kind of the answer. Strongly deleterious variants. Let's focus on that. Tibet has much fewer. The total number of strongly deleterious variants are fewer because the population size is smaller, and our models suggest that those variants can be purged out. Whereas in a large population, it's much more difficult to purge out those variants because they're all very rare, and the probability of them meeting up as a homozygotes is very unlikely. So large populations have more genetic load, something very well established in theory. And that means the founders of Isle Royale from this large population on the mainland brought in with them much more genetic load. And that genetic load was then expressed with inbreeding. So in a sense, the founders were contaminated with this genetic load that was then expressed by inbreeding. And we can model that whole process. I won't go into the details of a model, but there's this wonderful program called SLIM. It's a very complex model, just came out last year, which is like a population viability analysis wrapped into a genetic analysis. And it makes real life, it models real life situations instead of all the many assumptions of population genetic theory that are unrealistic, like no selection, no mutation, all the properties of the Hardy-Weinberg equilibrium, which are very unrealistic. And it models, has a real kind of relationship between the absolute fitness of individuals and the population size. And it has stochastic population dynamics and incorporates things like environmental stochasticity, natural catastrophes, natural random changes in population size. It's a sophisticated PVA, population viability analysis. Even the instruction manual is over 500 pages. So that's what we did. We modeled Isle Royale from an ancestral population size of 10,000 and then various source populations. And I just want to jump to the result here. We model it as 20,000 genes, which is about how many are in the wolf genome, 38 chromosomes. We give a mutation rate. And then here's the result. And it kind of reiterates what I've been trying to say. And that is, if we just assume that on Isle Royale, the population size, the carrying capacity was 25 versus 50 versus 100. And then this is the ancestral carrying capacity of the population where the founders were drawn. So this is the mainland population. You can see if we have 15,000 as the ancestral population size from which the founders were drawn, that the time to extinction is far less than if we draw those individuals from a population size that only has 1,000 individuals. That's because the 1,000 individuals have more opportunity to purge, there's more homozygosity being generated, and so they arrive with less of a genetic load. And the effect's quite dramatic, more like 400 generations versus less than 50. And similarly here and here, the smaller founding, the smaller size where the individuals are drawn kind of pre-adapts the founders for life at a very small population size. And this persists all the way through. This is the generations to extinction. We can even ask, well, how about we pick individuals with maximum heterozygosity, maximum variability, which is the paradigm today. We choose individuals or populations that have maximum amount of heterozygosity with the notion that they have the maximum spectrum of adaptive variation that they'll need to persist into the future. Well, that doesn't do as well because this population is generally large. However, if we pick individuals that have fewer deleterious variants, as we can categorize them, the population has a much larger persistence time. So genetic rescue is most effective when targeting moderate-sized, medium-sized source populations with low levels of deleterious variation. And we can pick those founders with modern genomic techniques. And the sweet spot seems to be about 1,000 individuals. Any less than that, we have problems with historic inbreeding. Any more than that, we have too much genetic load. And so it's even better than no rescue at all. No rescue at all is better than picking one from a large population. So extinction risks in small population, strongly influenced by ancestral demography. Just like we would consider the fossil record, ancestral demography is the history of populations as much as we can model it. So founding or augmenting small populations with individuals that have a high genetic diversity has counterintuitive effect of increasing extinction risks because there's more genetic load in those large populations. The more deleterious variants can accumulate and be hidden in the heterozygous condition. So the management decisions for the Isle Royale population and other populations that we want to alter on islands, we should focus instead on minimizing deleterious variation, the individuals we choose as founders, and we can do that just by taking a blood sample and looking at the genome, rather than the small population paradigm of maximizing genetic diversity. And I have to say, it's an add-on here. The Isle Royale wolf has been reintroduced from mainland, and we talked to them extensively about how best to do it. And unfortunately, they weren't interested in that. And they took individuals from 3 distinct populations. So actually maximizing the genetic load that was reintroduced into the population. I just want to finish here with some other species that you all might be interested in. Here in California, mountain lions, famous P22, the lonely bachelor in Griffith Park, taken in front of the Hollywood sign. Are they inbred? Or are mountain lions inbred? Turns out, yes, they are. And mountain lions suffer from isolation by roads, and also they're top predators, so their population sizes tend to be small, and also a single male generally mates with many females and defends those females across a wide area, so their effective size is even smaller. Here are the canonical cypress swamp mountain lions, and here's their level of heterozygosity, extremely low. They stopped reproducing. They no longer had viable sperm and could not reproduce. They're cryptorchids. And so here's their level. How about the Santa Monica Mountains? Here's our level of homozygosity, runs of homozygosity. There's another individual different, but we're approaching that level. In fact, now there's been a report of cryptorchids in the Santa Monica Mountains and the kinked tail, which is indicative of inbreeding, and the Florida population. But here was our surprise. Here are mountain lions from the Santa Cruz area. We actually took those initially as our outgroup. And we thought, that should have high levels of variability, but you can see the proportion of ROH is actually very high. And here's Yellowstone National Park. That was our other outgroup. But look across the Yellowstone National Park genome. There are large areas where there's no variation coming from inbreeding. Only when we get out to Brazilian mountain lions do we see a healthy genome with no inbreeding. There's one spot where there's no evidence of inbreeding. So we're beginning to conclude that all across California, probably mountain lions are intensely inbred and on the verge of inbreeding depression to an extent which they may not be able to breed. The last species I'm going to mention is the California sea otter. This is work done by Annabelle Beichman in my lab. She's now a postdoc at University of Washington. And we can ask the same question about sea otters, whether they're intensely inbred and whether they're likely to suffer genetic deterioration. And she used a capture array, and we did a study across the entire geographic range. And turns out there's geographic units, the ones in Southern California and British Columbia, where they've been translocated, very distinct from everything else, where there is kind of a geographic pattern from Alaska all the way across to the Commander Islands. And this is just a structure classification, if you will, of individuals which represent each line. And you can see Alaska, the Commander Islands, the Kuril Islands, all very distinct. This is actually Aleutian Islands, I'm sorry, Commander Islands. Alaska, there's some evidence of admixture. California, no evidence of admixture. And this just shows even historic specimens. We've had some wonderful ancient sea otters that are over 1,000 years old. And so the ancient specimens from California group with modern day California sea otters. And that really suggests that there's been continuity at least more than 1,000 years. In the same way, ancient California sea otters are classified with modern California sea otters, different from Alaska and Aleutian Islands. So we did the same kind of modeling. In this case, what's happened to sea otters is a very recent, like a few generations ago, devastation due to the first trade. We call that a two-epoch model. So they had an ancestral size and then a bottleneck and then reached a current size. And I just want to show you these results quickly. There has been a remarkable decline in population size of sea otters from an ancestral size in California to today's numbers, which are 6% of what occurred historically. And you can see across the board, all these sea otter populations independently survived this kind of bottleneck, which to us means that really how sea otters recovered was not from one refugia, two refugia, but they recovered from multiple refugia and repopulated their geographic range, preserving their geographic structure and presumably some of the local adaptations that might have occurred there. Okay, I'm not going to go into the load stuff, but I will just say that it seems genetic load that has accumulated in sea otters is not a threat. That is, the genetic load that accumulated here under various scenarios, demographic scenarios with the bottleneck, will decline with time and eventually reach historic levels. But it's not a serious increase in genetic load over time. In fact, we can say, will this genetic load get in the way of recovery? And so here's the recovery, the bottleneck, and then this is the genetic load. It's high right now, but eventually during the recovery period, it's going to decrease like I was just showing to you, and it will continue to decrease. We can halt that decrease by augmentation with individuals from elsewhere, but actually it's not particularly needed. There's no big threat with regard to recessive alleles. So uniform decrease across the geographic range, independent recovery, structure still accords with geography because of that. Low population sizes prior to the fur trade, actually they were not that abundant. Catastrophic decline, elevation of genetic load, expansion in the future result in small decline in genetic load, no genetic threat, and translocations may be beneficial. Okay, I'm just going to give a call out before I end to some of the programs that are out there. There's CalEDNA, and this is one of our big pushes now, which actually uses environmental DNA to tabulate diversity of communities all across California. And we have some great colleagues at the Natural History Museum who are helping us and us interested in the marine environment as well. And one big project that has been developed by Brad Schaefer, who's the PI, was complete genome sequencing of 150 species, most of which are endangered from throughout California, and complete genomes of 150 individuals from each of these species. This is something that was funded by the state for $12 million and has a long list of PIs. I encourage you to take a look, but more analyses, as I've described, are going to be done on California species, from plants to marines species to vertebrates. Also, the other big initiative, and I'm just going to end here, is the Earth Biogenome Project, still getting off the ground. That is to sequence everything from a few places and try to understand the interactions of adaptation, demography of a whole community from one place. Okay, I think we're going to end there. I want to thank you all very much. It's delightful. I hope it wasn't overwhelming, but hope to see you actually in person sometime.
Great. Thank you so much, Bob, for that presentation. There'll be roaring applause. You can just kind of imagine in your head.
Oh, someone's trying to call me.
Anybody has any questions, please put them in the Q&A or in the chat. I will be happy to read questions from both of those. I found your talk just fascinating. You know, a lot of that is what I'm interested in. One question I have for you, just while we're waiting for any others, is, you know, so many of introduced species that we see come from species that have historically very large effective population sizes. You know, I'm thinking about, for example, the many non-native birds that we have here in LA, or insects or things like that. What do you think makes those species so successful, given that they probably have very small numbers of founding individuals and come from these large effective population sizes? I guess, you know, this is kind of a question that I would like to actually look at and address, but do you think it has to do with the gene flow or just minimum numbers of founding individuals? What are your thoughts there?
It is true that many of the introduced species come from large populations, and then their introductions are very small, but they probably increase very quickly. So if they're very successful, and most introductions are not successful, the ones that are increase very quickly. So they, in a sense, preserve much of the genetic variability that's there. And then they're large population size, so they don't have an opportunity to inbreed and express all of this deleterious variation. If they remained at very small population size for a long period of time, then you would expect inbreeding depression. But I think that the trick is to expand your numbers very quickly so that these rare alleles never manifest as a homozygote and cause inbreeding depression.
Makes a lot of sense. Thanks. We've got another question from Greg Polly. He says, thanks for that fun presentation. Why do you think that any of San Nick is so much lower than Miguel, given that Nick is 50% larger island?
I don't know. I mean, I think because there's a demographic overlay there, there may have been many bottlenecks over time that we're not seeing, but I'm not sure. I mean, I'd expected San Miguel was going to have extremely low variation, and they are low, but not quite as low as San Miguel. So it may be there were a number of historical bottlenecks. We know going back 100 years, there's no evidence of an additional bottleneck, but it may be before that there were. And Native Americans may have augmented the situation by their activities. I mean, the Native Americans had dogs on these islands. I can't imagine how they all got along.
Great. We have one more question, which is, has similar work been done, and this is from Patrick, has similar work been done for small isolated human populations?
Oh, there's a huge literature on human populations. The most notable one is the Icelandic human population that had a narrow founding. In fact, they've made like a whole kind of industry of it. There's biotech companies that are actually sort of mining the Icelandic genome, looking for deleterious variants, how they've been expressed, how that affects longevity. And they have, because of Iceland, what it is, they have actually complete medical data on just about everybody. And they actually know how they're related. And they have identified many deleterious variants that have been preserved in that population. But likewise, other isolated populations worldwide have been studied. And the literature is really dense and wonderful. I mean, humans have gone from like a laughable species for scientific study because you can't do experimental manipulations to the leading species for understanding genes and phenotypes because there's so much data on humans and so many humans, hundreds of thousands of individuals have their genomes sequenced.
Thanks for that. So we're a little bit short on time. We have one more question, which I'm going to take this from Regina. Regina asks, would we expect this is the same for vertebrates and invertebrates? For example, where most invertebrates have short life cycles?
Hi, Regina. No, I think there's going to be a dramatically different pattern. I'm sorry, the cat's eating my cookie. Come on, let's go. Get out. And it's hard to imagine, except in an island situation, what might be happening with insects. It may be across the trophic pyramid where the predators, the top predators of insects, as insects, have much lower genetic variation, and in some situations where they maybe founded a new population and haven't increased in numbers very quickly, that inbreeding depression is manifest. I mean, there's been wonderful studies on like some butterflies actually by Ilka Hensky and his colleagues that actually show some of the genetic effects of isolation on a landscape and small population size in terms of the expression of deleterious variation. Ilk Sakurai is one of the people working on that issue in a metapopulation sense.
Great. Thanks for that answer, Bob. And since it is 1 o'clock, I'm going to say we should all virtually applause and thank Bob. And if you have any other questions, as you know, Bob said you could email him, but thank you so much for joining us today. It's really been a pleasure having you. And I look forward to seeing more of your work that comes out.
Okay, thank you. I appreciate it.
Thank you, Bob. It's a wonderful talk.